Very good evening. So today we are going to start with the next important topic in your molecular biology after completing your purine and pyrimidine metabolism. Now we are going to move towards a very important topic that is going to be your basic structure of your DNA and followed by your DNA replication. So before moving into the basic structure of DNA, what I wanted to just revise is, you know that your DNA or your RNA, it is going to be formed by nucleotide and phosphate bases. So nucleotide is going to be formed by nucleoside and phosphate. Nucleoside is going to be formed by nitrogenous base and your ribose sugar. Nitrogenous base is again made up of purine and pyrimidine. This is what we discussed, and if you have any doubt in this, you can just put it in the chat box. Now, with this base, let's move into the next one, that is going to be your DNA. Okay, so DNA, if you're going to tell me, DNA is nothing but it is a polymer of deoxyribose, which is linked with by the 3' to 5' phosphodiester linkage. You already know that your phosphodiesters are the one that links the one nucleotide with the other nucleotide. Okay, so it is a 3' and the 5' வரைக்கும் ஒரு ஸ்ட்ரெயிட்டா இருக்கக்கூடிய ஒரு செயின். அப்ப இது வந்து என்னன்னா, ஒரு சென்ட்ரல் டோக்மால you're going to have your chromosome. இந்த chromosomes நீங்க unwind பண்ணீங்கன்னா, so if you're going to unwind this chromosome, what you're going to get is a single-stranded DNA. So upon the single-stranded DNA, bead-like appearance, it is going to be packed in such a way that they are folded, and they are formed chromatin. And this chromatin forms your chromosome. In the basic idea, I want you all to know. Followed by it, so now you have to understand the basic structure of your DNA. To understand the basic structure of your DNA, what you have to know is the DNA oda structure first who gave it like a double helical structure. So double helical and the இப்ப இருக்கக்கூடிய அந்த terminology of why we call it as a double helical right-handed helix அப்படி நம்ம சொல்றோம்ல, இத நேம் பண்ணவர் யார் அப்படின்னு பார்த்தோம்னா வாட்சன் அண்ட் கிரிக். ஏன்னா he is the one who demonstrated it. Okay, who is the one who demonstrated the three-dimensional structure of your DNA. That is the reason why he got Nobel Prize. Now, if you ask me, is he the first person to demonstrate? No, you had a person who demonstrated in the X-ray crystallogram for the first time and he named it as DNA. He is none other than Rosalind Franklin. So Rosalind Franklin அப்படிங்கிறவர் தான் first DNA demonstrate பண்ணாரு X-ray crystallography-ல. அப்புறம் you had this Morris Wilkin who also gave contribution for the DNA's structure. But then ஒரு ப்ராப்பரான rules first time வந்ததுன்னா அது after this formation of this three-dimensional model. அதனால தான் வாட்சன் அண்ட் கிரிக்கு we awarded Nobel Prize. So when you're going to just look into this, can you appreciate? So this is going to be somewhat right-handed double helix. So they are going to be right-handed double helix. It starts from your 5' and ends in your 3'. There is going to be 2 n, and each strand is going to be anti-parallel to each other. That means that one strand will be going 5' to 3' and other strand going 3' to 5'. They won't be going in a straight line, and that's why you call it as anti-parallel in nature. And there came the Chargaff rule. So if you wanted to just understand the structure of DNA, you need to identify the Chargaff's rule. Just listen to me carefully, then I will show you. You can copy down all the rules. So what is your Chargaff rule tells us? Number of purines should be equal to number of pyrimidines. That is the first thing he is telling. Number of purines should be equal to number of pyrimidines, and the entire DNA has to be made up of only purine and pyrimidine, not any other structure apart from it. So that is the second rule. And third rule is, you have a purine and you have a pyrimidine. Between this purine and pyrimidine, there must be a hydrogen bond. So this hydrogen bond must be, okay, when between your adenine and thymine, can you see between adenine and thymine, what you have is a double hydrogen bond. Whereas between your guanine and cytosine, you are going to have a triple hydrogen bond. So your number of purines should be equal to number of pyrimidines, made up of only purine and pyrimidine. Third one is going to be, it is made up of hydrogen bond. Between your adenine and thymine, you have double bond. Between your guanine and cytosine, you have triple bond. And this is going to be called as your Chargaff rule. Remember, Chargaff rule is only applicable for DNA. It is not applicable for RNA. It is not applicable for single-stranded DNA. Okay, it is not applicable for even single-stranded DNA. It is applicable only for double-stranded DNA. And I want you all to remember this one particular thing. Second thing, after knowing this, next important thing what you're going to know is, Okay, after following all these things, what you are going to know is, you have to know that this is the Watson and Crick. This is going to be the B form of DNA. Now, B form of DNA, if I tell, what are the different forms of DNA you have? So you have three different forms of DNA actually. So the DNA is going to be made up of, is going to be of three types. Number one, it is going to be A type, B type, and Z type. A type, B type, and Z type. So A, B, Z. So A is usually present in the animals. A for animal. A usually present in animal. B is usually present in the humans, and Z is usually present in the fungus. Z is usually present in the fungus, and A has 11 base pair per turn, whereas B has 10 base pair per turn, and Z has 12 base pair per turn. And then the pitch or number of base pairs in one pair. Okay, number of base pairs, on the base pairs or the length எவ்வளோன்னு நம்ம பார்த்தோம்னா, it is going to be around 0.26 for animals, 0.34 for human, and 0.57 for the Z for your what to say, fungus. And next important thing what you need to understand is, in humans it is going to be right sided helix. In animals also it is going to be right-handed helix, whereas in fungus it is going to be left-handed helix. So this is what I want you all to know. And next important thing what I want you all to know is the organic structure of your DNA. So what happens is, whenever I am going to just break down your DNA, whenever I'm going to just remove your DNA, I find the pitch length is going to be 3.4 nanometer. Each turn of your helix is 3.4 nanometer. And each turn has 10 helix, 10 group of base pairs, and each base pairs are at the distance of 3.4 Armstrong. So 3.4 Armstrong. So 3.4 Armstrong is nothing but 0.34 nanometer. 0.34 nanometer. Okay, is it clear, guys? 0.34 nanometer. And the width of each helix is around 20 Armstrong. Its width of helix is around 20 Armstrong. So a single term of bDNA contains 10 base pair. That's what we told. And the major groove has a bond more than 180 degree, and minor groove has bond less than 180 degree. Okay, so this is the important. And when we are going to denature the DNA, and when you're going to analyze the structure, so what we find is, okay, what we find after denaturation is, only DNA rich is in the GC pair melts at higher temperature when compared to the AT pair. Why? Because triple bond and double bond is placed over them respectively. The strands of DNA separate over temperature range. So the midpoint is called as melting temperature. What is TM or melting temperature? It is going to be the place where the 50% DNA gets denatured, and this depends upon the triple bond. If it is going to be between guanine and cytosine, which has a triple bond, then it is going to be having a more melting point compared to that of the double bond between your adenine and thymine. Okay, so what you are going to use is, you are going to use a formamide, which is used for studying the DNA denaturation. Okay, this is your Z form of protein. Z form of your DNA is going to be found in your fungus. Okay, so this comes to the end. I want you all to copy this down. Once you copy this down, I will move on to next one. So kindly pause this. So it's a right-handed double helix. Polarity between 5' end to 3' end. It is anti-parallel. Chargaff's law. Purines pyrimidines தான் இருக்கும். Purines is equal to pyrimidines. And then purine, between your purine and pyrimidine, there is a hydrogen bond. Between your adenine thymine, double bond. Between your guanine and cytosine, is going to be triple bond. Okay, this is what we discussed till now in the first 10 minutes. Next, we are going to move towards a very important topic, that is going to be your histones. So what is a histones? Just listen carefully, guys. So you have your chromosome, isn't it? So that chromosome, when you're going to unfold it, when you're going to unpackage it, so you can see chromatin. And when this chromatin are slowly unbounded, you are going to have your DNA. Okay, so that means that your DNA are packed in such a way that they are together, they are folded, and they form this chromosome. Okay, they form this chromosome. So then what you need to understand is, who is going to help in the folding of this DNA? Who is going to help in the folding of this DNA? The person who helps in folding of your DNA, he is none other than histone. He is none other than histone. That is what we are going to see now. So can you see in this picture, guys? You have this DNA. DNA is going to be wrapped around. He is going to be wrapped around by histone. So what is this histone? These histones are nothing but they are going to be someone who is a positively charged. So in a simpler term, if you want me to tell, your DNA, so in a simpler term, if you want me to tell, your DNA are negatively charged. They are negatively charged. They are acidic in nature. So they are interested to pair up with someone who is going to be who are positively charged. So that's where your histone comes into play. Histones are positively charged. Histones are positively charged. So they are going to have a force of attraction between each other. So you have four types of histone basically: H1, H2, H3, H4. H2 is divided into H2A, H2B. So usually what happens, there is going to be a cubical formation by H2A, H2B, H3 and H4, upon which you have your DNA wrapping around. So you have your DNA wrapping around the histones, and the two ends of your DNA are going to be attached to another or linked to another histone, that is your H1. So that is the reason why H1 is called as linker DNA. That is why H1 is called as linker DNA. What is the significance of this? This histone creates a modification in your DNA. This helps in the promoting the transcription in the gene. It is going to promote the transcription in that DNA. So who is going to do that? That is going to be done by your histones only. So what are the different types of histone? Before moving to that, what you need to understand is, what is the ultimate aim of your histones? The histones ultimately pack your DNA to form chromatin. So this chromatin are of two types: euchromatin, heterochromatin. So this chromatin is of two types. What are the two types of chromatin? You have the euchromatin and heterochromatin. Euchromatin is activation of your transcription, whereas heterochromatin acts by inhibiting your transcription or inactivating your transcription. So what happens is, this histones are going to cause some modifications. So what are those modifications? I usually remember it as Miss Rupa or Rupa MS. So R stands for your ribosylation, U stands for ubiquitination, and P stands for your phosphorylation, A stands for acetylation, M stands for methylation, and S stands for sumarization. Okay, sumarization. So இதுல வந்து நீங்க எது நல்லா நான் வச்சுக்கணும்னா, உங்களோட ubiquitination, it reduces the transcription, whereas again sumarization, it is going to decrease the transcription. And you have basically two important things here, that is going to be your methylation, acetylation. So your acetylation, what happens is, it is going to cause acetylation is going to cause increased activation of your transcription. Okay. So, okay. So, I think you have a doubt. I'll tell you again. So, histones are nothing but they are the one where your DNA are going to wrap around each other. They are going to be controlling your transcription. They are going to be controlling the DNA to move into a replication or transcription. So what happens here basically is that the histones gets modified or there are some modifications occurring in the histone. So that is called as certain names called as Rupa MS. R stands for your ribosylation, and U stands for ubiquitination, P stands for phosphorylation, A stands for acetylation, M stands for methylation, and S stands for sumarization. So idila ubiquitination reduces the transcription phase, and whereas again you have your sumarization, which will be reducing the transcription phase. Apart from it, you have two important things: acetylation, methylation. Acetylation causes increase in the activation of your transcription. That is going to be it's going to be euchromatin in nature. Whereas your the other one, that is going to be called as your methylation, is going to be heterochromatic. Heterochromatic means they inactivate your transcription. They are going to cause inactivation of transcription, and this is what they are telling. So this is going to be called as your histone modification. Okay, so this is called as histone modification. What is going to be euchromatin? Euchromatin means uncondensed chromatin. So chromatin is open to give DNA. Whereas what happens in heterochromatin, they are going to be not uncondensed. So ultava, they are going to be condensed because they are going to be condensed. So transcription is going to be inactive in this. So transcription is going to be inactive in this. I hope you can understand that. Next, what happened is, who did all these things? So from which one we got all this information? All these we got from Human Genome Project. So Human Genome Project is a funded project by several societies to identify the genetic material. What they did is they studied the entire genomic sequence. Okay, they then studied the entire genomic sequence and gave a shocking fact that when you're going to stretch your genome, stretch your DNA. If you want to stretch the DNA of the one particular person, you can extend from the in the earth to moon. So அந்த அளவுக்கு பெருசா இருக்கும் human genome project சொன்னாங்க. But அவ்வளோ பெருசா இருந்தாலும் அதுல 98% are going to be junk in nature. Introns, okay, you have introns, you have your reverse transcriptase, pseudogenes, everything, they are going to be junk in nature. Who is going to be helping in nature for us? The only 2% of expressed genetic information is going to be helpful, and the expressed genetic material are going to be in the form of DNA. So it is going to be in the form of DNA. Now you understood why DNA is important. Okay, now you understood what are going to be the different process of your DNA. Remember, the DNA is going to be binded together by the structure called as histone. What is the other structure? So can you see in this picture? I'll show you the picture again. Can you see in this picture? You have your histones, and apart from it, you have another small round. What is that? That is called your motifs. So you have your histones and motifs who are going to bind your DNA and keep it together. And that is what we want to implicate with this chapter, the NMC competency, which is a 7.1 DNA organization and structure. I hope I have completed the very important topic. Next, we are going to deal with a very important topic. Are you all ready? Cell cycle and DNA replication is what we are going to see. This is again a very important topic, DNA replication. What I will do is, I have a notes also for this. So what I have is, I have two important things for you. One I have as a notes, and other I have as a PPT. So I will explain you as a PPT, then we will revise it with the help of the notes which I have. So this is the notes. I will send you the notes also, don't worry. First we will discuss in detail, then we will see the notes. Okay, so it will be helpful for you to understand. Once you understand the cell cycle, then you go for the DNA replication. So clear, guys? Can we move forward? Hope so. You're all very clear with the three topics which we have discussed till now. That is going to be a transcription. That is going to be your that's going to be your nucleotide chemistry. Then we moved on towards the nucleotide chemistry. Then we moved on towards your cell cycle. Now we are going to move towards your cell cycle. So clear, guys? So first, let me start with your cell cycle. So remember, so cell cycle, you all can, actually, I hope so you all remember, you have a cell cycle like this. G1 phase, S phase, then you have G2 phase, then you have your M phase. This is going to again move into G1 phase. So you have three arrests. So one, there can be arrest at your G1 arrest. So that is going to be called as your G1 arrest. Okay, there can be arrest at this particular point. There can be arrest at this particular point, G2 arrest, and there can be arrest at the M stage. There can be arrest at M. Okay, so let's go one by one. Why do you need this cell cycle? Why do you need this cell cycle? Cell cycle is nothing but just put the heading as cell cycle. Why do you need a cell cycle? A cell cycle is needed for organized events. Okay, it's organized events that leads to multiplication of cell. Okay, so that leads to multiplication. So pluripotent stem cell நம்ம உருவாகி அதுக்கப்புறம் நம்ம வந்து multicellular organism நம்ம வளர்ந்துருக்கோம்னா அதுக்கு okay, you need the multiplication. You need multiplication of cells, isn't it? You need multiplication of cells. So that cells should be organized event, programmed event. Okay, they are going to be helping in the growth. Okay, they are going to be helping in the reproduction. Okay, they are going to be helping in the wound healing. But unfortunately, along with this all good things, there is one bad thing of this also. The bad thing is nothing but, what is going to be the bad thing that we have along with this? The fourth one, that is going to be your cancer. So cancer இங்கேருந்தா வருது. ஸோ இந்த செல் சைக்கிள்னால தான் கேன்சரும் வருது. அப்போ நார்மலாகவே கேன்சர் வரணும்ல எல்லாருக்கும், ஏன் வராமல் இருக்கு அப்படிங்கிறது ரொம்ப இம்பார்ட்டன்ட். ஸோ we are going to consider this. Okay, you are going to have your cytoplasm, you are going to have your G0 phase. Along after G0, there's going to be G1. Then you are going to have your S phase. After S phase, you're going to have your G2 phase. Then you're going to have your M phase. Then you have your cytoplasmic division. Then you have cytoplasmic division. So, in the mitotic phase, it is going to be your M and your மீதி M phase மட்டும்தான். இந்த M phase மட்டும்தான் you called as mitotic phase. Okay, மீதி இருக்கிற எல்லாமே it is going to be all these things, they are going to be interface, they are going to be nothing but they are going to be interface. Okay, மீதி எல்லாருமே interface தான். புரியுதுங்களா? இது வரைக்கும் ஏதாவது டவுட் இருக்கா இது வரைக்கும்? Okay, the first step, what happens is there is an accumulation of raw material. Now what you need, so if you wanted to multiply something, if for example if you wanted to reproduce the idli what you made, okay, for reproducing idli what you made, what you want, first you want to know the recipe from it. So first you want to know the recipe from it, then what you want to do, you wanted to know what are the raw materials that are needed. So you have to first, what you need to do, you have to accumulate the raw material, that is your G phase. Accumulate your raw material, and that is going to be your G phase. After G phase, what happens? You have this S phase. S phase la enna pandrona, you are going to go for replication. Okay, you're going to go for replication. First, you're going to accumulate the raw material. After accumulating the raw material, what you're going to do? You're going to go for replication. Okay, after replicating, what you do? So, okay, if I replicate panta, so if I accumulate raw material, accumulate panta, if I replication panirken, if I the replication noda and the idea. Okay, so உனக்கு என்னென்ன தேவை? replication na இட்லி மாவு பண்ணிட்டேன். So first இட்லிக்கு தேவையான பொருளை ரெடி பண்றது G phase. எஸ் பேஸ்ல இட்லி மாவு அதே மாதிரியே நான் அரைச்சிட்டேன். இப்ப இட்லி மாவு அரைச்சா மட்டும் போதாது, அதே மாதிரி உங்களுக்கு என்ன வேணும்? அந்த ஷேப்ல இட்லி கிண்ணம் வேணும், பாத்திரம் வேணும் ஃபார் புட்டிங் தட் இட்லி அண்ட் ஸ்டீமிங் இட். ஸோ அப்ப அதுதான் வந்து உங்க G2 phase. G2 phaseல என்ன பண்ணும்? டூப்ளிகேஷன் ஆஃப் அதர் ஆர்கனல்ஸ். ஓகே, ஆஃப் அதர் ஆர்கனல்ஸ். So okay, once you duplicate the other organelles, finally what happens is நீ அந்த பாத்திரத்துல மாவு ஊத்தி குக்கர்ல வச்சு you are going to wait till it steams and comes out. So அந்த steaming phase தான் M phase. இங்க என்ன ஆகும்? உங்க எப்படி உங்க இட்லி மாவெல்லாம் பாண்ட் ஆகி ஒரு சாலிட் இட்லி கிடைக்குதோ அது மாதிரி it is going to have a spindles. It is going to form a spindle and it is going to form a solid structures. So spindles formation. Okay, understood. So these are the four important G, S, G2 and M. This is what you need to understand. So if this cycle has to occur, what happens is a very important thing what you need to understand here is you need to understand that there must be some checkpoints to manage it. So நம்ம வந்து இந்த ஒவ்வொரு இடத்துலயும் நம்ம கண்காணிக்கணும், ஒவ்வொரு இடத்துலயும் நம்ம செக் பாயிண்ட் வைக்கணும். அப்படி இல்லைன்னா இட்லி மாவு நல்லா வராது, இட்லி மாவு நல்லா வரலன்னா நான் இட்லி நல்லா வராது. அதே மாதிரி பாத்திரம், இட்லி பாத்திரம் கரெக்டா இல்லைன்னா இட்லி நல்லா வராது. ஸோ இந்த மாதிரி உங்களுக்கு ஒரு ஃபுட்டுக்கே இவ்வளோ செக் பாயிண்ட் வைக்கணும்னா நம்மளோட டிஎன்ஏ ப்ரொடக்ஷனுக்கு நம்ம பாடி செக் பாயிண்ட்ஸ் வைக்கணும். அப்ப அந்த செக் பாயிண்ட் யார் ரெகுலேட் பண்றாங்க அப்படின்னா சிடிகே. Who is that CDK? What is CDK? Cyclic dependent kinase. Cyclic dependent kinase is the one that is going to regulate. That is very important. So you have your cyclin. Okay, what are the different cyclins? You are going to have your, okay, you have cyclin A, B, E. So you have cyclin A, B, E. Okay, you're going to have your G1 S phase and G2 M phase. Okay, you have two important phases. So what are the two important phases? You have G1 S phase and you have your G2 M phase. G1 S phase and G2 M phase. So these are the two checkpoints you need to understand. These are the two checkpoints which you needed to understand. So after understanding these checkpoints, these cyclic dependent kinase, you have inherently you have two important gene in your body. One is going to be your proto-oncogene, and other is going to be your tumor suppressor gene. Other is going to be tumor suppressor gene. So one is going to be your proto-oncogene, other is going to be your tumor suppressor gene. What is a proto-oncogene? So proto-oncogene is something that is going to be having a gain of function mutation. So what happens? So when the proto-oncogene, when it is going to have a gain of function mutation because of some X-ray, ultraviolet rays, radiation, it is going to form oncogene, and this oncogene forms oncoprotein, and this oncoprotein is going to cause cancer. Okay, similarly you're going to have your tumor suppressor gene. When this tumor suppressor gene attains a loss of function mutation, this leads to cancer. We have certain example for both of them. So for this example is going to be a FOS gene, MYC gene, you have your JUN gene, then you're going to have BRCA1. Okay, then tumor suppressor gene, again you're going to have your P53. Okay, you're going to have RB gene, you're going to have retinoblastoma gene, you're going to have P53 gene, then you're going to have your retinoblastoma gene is going to form Lee-Fraumeni syndrome. Lee-Fraumeni syndrome. It is going to form Lee-Fraumeni syndrome. Okay, which one? When there is a P53 mutation. Similarly, retinoblastoma mutation causes Wilms tumor. It is going to cause Wilms tumor. Okay. This is what I want you all to understand, guys. So once you understand this, before moving into the DNA replication, so this is your cell cycle. So I hope so you can understand your cell cycle. Cell cycle more than enough. So again I repeat you have 4 phase. G1 phase. G1 phase raw material. So raw material it is the phase where you have the protein synthesis, RNA and protein synthesis. Once you have this RNA and protein synthesis, there is going to be a DNA RNA synthesis. Okay, DNA RNA production. The DNA RNA production G2 in the DNA RNA production it has to again replicate. It is going to duplicate it and form it arrangement, tubulin arrangements. Other mitosis it is going to divide. It is going to be dividing, and then you have your cytokinesis. You have a G0 phase. What is G0 phase? G0 phase is nothing but they are the resting phase. Okay, non-dividing phase. They are going to be called as your non-dividing phase. Non-dividing phase. Okay, this is going to be called as your resting phase. This is going to be called as your resting phase. Okay, what you can actually do is you can arrest at G1, you can arrest at G2, or you can arrest at mitotic stage if there is any damage caused. Or damage in the stages we can block it. Okay, and the madri block panla, and you can give external drug to block certain areas. So and the block pandra first one G2 phase of block pandra drugs. Okay, now we are going to identify and write what are the drugs which are going to be blocking your different phases. G2 phase of block pandra drug 5-fluorouracil. You have your 5-fluorouracil and you have your methotrexate. Okay, you have your 5-fluorouracil and methotrexate. So in the 5-fluorouracil methotrexate, they are thymidylate synthase inhibitor, or DMFR. So it's going to inhibit your dimethyl reductase, folate reductase. So in the stage inhibit pannu. So you're going to have your colchicine, paclitaxel, Wincop alkaloids. They are going to block your mitotic spindle formation. So you have your colchicine, isn't it? So colchicine blocks your mitotic spindle formation, and you have your tubulin formation in your G2 phase. So you have your tubulin formation in your G2 phase. Okay, you have your tubulin formation in your G2 phase. This tubulin formation is going to be inhibited by bleomycin. It is inhibited by bleomycin or etoposide. So that is what I want you to understand at this particular point. Next is going to be, we are going to discuss about the DNA replication. So to start with this DNA replication, come on guys, to start with the DNA replication, what you need to understand is you have 2 important things. That is going to be, one is going to be a prokaryote and other is going to be eukaryote. So prokaryotic DNA ko eukaryotic DNA ko sila difference irukku. Okay, prokaryotic DNA ko eukaryotic DNA ko sila difference irukku, adha nama paaka porom. Okay, before going into those things, you have to identify the origin of replication. Okay, then unwind your DNA, formation of replication for primer synthesis and primer removal and ligation of new DNA segments. So this is the entire thing what we are going to form and how we are going to repair everything we will be seeing now. Are you ready, guys? So what is the DNA replication? The DNA replication is a process of copying and duplicating the DNA molecule. Move on towards this DNA replication. So in the DNA replication, main duplicate panna poranga. And the duplicate create பண்றவங்க பேர் தான் வந்து DNA polymerase. So இந்த ஒரு காம்பவுண்ட் தான் நமக்கு ரொம்ப இம்பார்ட்டன்ட். DNA polymerase இவங்க தான் ரொம்ப இம்பார்ட்டன்ட். They are going to be playing a major role for DNA replication. So DNA replication na என்ன? Simple. Okay, DNA replication na enna? So you have a one strand that is going to be 3' to 5'. Okay, then you have other strand that is going to be running anti-parallelly 5' to 3'. கரெக்டா? ஸோ இப்போ நம்ம என்ன பண்றோம்னா இன்னொரு ஒரு கிரியேட் பண்றோம். இதே மாதிரி இதோட ஒரு டூப்ளிகேட்டா கிரியேட் பண்றோம். அப்ப டூப்ளிகேட்டா கிரியேட் பண்றப்ப அது எந்த டைரக்ஷன்ல இருக்கும்? கரெக்ட் தானே அந்த கேள்வி. ஸோ இது வந்து 3' to 5' இருந்ததுன்னா இதுக்கு ஆப்போசிட்டா கிரியேட் ஆகிறது வந்து என்ன இதுல இருக்கும்னா ஆப்போசிட்டா இருக்கும் 5' to 3' இருக்கும். சிமிலர்லி 5' to 3' என்ன ஆகும்? திஸ் ஃபார்ம்ஸ் 3' to 5'. ஸோ உனக்கு அப்ப ரெண்டும் வந்து பாத்தீங்கன்னா ரெண்டு பேரும் they are going to replicate. அதை யார் replicate பண்றாங்க? DNA polymerase. அப்ப you have two important group of people, eukaryote, prokaryote. So you know that in prokaryote the DNA structure is different, in eukaryote DNA structure is different. அப்ப DNA polymerase who is going to form and who is going to help in this process is also going to differ. So அதுதான் நம்ம first பார்க்கிறோம். So first what you are going to remember is just find a empty space. The empty space, kindly write it down. Okay, so kindly write it down here. So you have some prokaryotic DNA and you have eukaryotic DNA. So prokaryotic DNA க்கும் eukaryotic DNA க்கும் என்ன difference அப்படின்னு நம்ம பார்த்தோம்னா prokaryotic DNA is going to be created by DNA polymerase 1, polymerase 1, polymerase 2, polymerase 3. ஸோ உனக்கு 3 DNA பாலிமரேஸ் இருக்கு. DNA பாலிமரேஸ் 1 என்ன பண்ணோம்னா பிரைமர் ரிமூவல், கேப் ஃபில்லிங், ரிப்பேர். ஸோ இதெல்லாம் நான் என்னன்னு சொல்றேன். சிமிலர் DNA பாலிமரேஸ் 2 மெயினா வந்து DNA ரிப்பேர் அண்ட் ப்ரூஃப் ரீடிங், அண்ட் DNA பாலிமரேஸ் 3 ஆர் கோயிங் டு ஹெல்ப் இன் ப்ராசஸிங் த ஸ்ட்ராண்ட். ஸோ எம்டி ஸ்ட்ராண்டை சிந்தசிஸ் பண்ணி ரீட் பண்ணும். ஸோ இது மூணுமே ஐ டெல் யூ வாட் இட் இஸ். பட் உங்களோட யூகேரியாட்டிக் டிஎன்ஏல இந்த மாதிரி பாலிமரேஸ் 1, 2, 3 எல்லாம் கிடையாது. ஒரே டிஎன்ஏ பாலிமரேஸ் தான். ஆனா அந்த ஒரு டிஎன்ஏ பாலிமரேஸ்லயே உனக்கு நிறைய காம்போனன்ட்ஸ் இருக்கு. வாட் ஆர் த டிஃபரெண்ட் காம்போனன்ட்ஸ்? யூ ஹாவ் யுவர் ஆல்பா காம்போனன்ட், யூ ஹாவ் பீட்டா காம்போனன்ட், யூ ஹாவ் காமா காம்போனன்ட், யூ ஹாவ் டெல்டா காம்போனன்ட், and you are going to have epsilon component. Alpha component is going to be acting as primase. So beta component is going to be DNA repairing, and your gamma component is going to be mitochondrial DNA synthesis, and your delta is going to form leading fragment and lagging fragment. What is this leading, lagging? I will explain you. And finally you have epsilon. Epsilon is nothing but they are going to be doing a proofreading. And this is what I want you all to remember. Once you remember all these things, okay, next let's go into it. Okay, so what are the components of your DNA replication? All happens from 3' to 5'. உங்களுக்கு என்ன தெரியணும்னா எல்லாமே happens from 3' to 5', except your proofreading. Everything is going to act from 3' to 5'. Except your proofreading, everything is going to act from 3' to 5'. 3' to 5' ஆக்ட் பண்ணாது ஒன்னே ஒண்ணுதான், அது வந்து proofreading. That is going to be from 5' to 3'. That is going to from your 5' to 3'. இது number 1. So first thing, okay, write it down along with me. What are the steps which you are going to remember? So write the heading as components of DNA replication. What are the components of your DNA replication? Number 1, okay, number 1 is all happens from 3' to 5' except your proofreading. So proofreading, which is going to act from 5' to 3'. Everything acts from 3' to 5' except your proofreading, which is going to act 5' to 3'. This is the first one. Second thing what you need to remember is you have something called as DNA ligase. So what is this DNA ligase? This DNA ligase na enna pannuna, it is going to anneal. You are going to anneal your DNA. That is the one that is going to anneal your DNA. Copy it down, I'll explain you later. Anneal your DNA. And third thing what you have to know is you have many structures called as, okay, one by one let's see. First you are going to have helicase. What is helicase? Helicase is the one, so you have two DNA, you have one DNA pa, like this. So you have your double stranded DNA. Okay, just remember like this. So you're going to have a double stranded DNA like this. What you have to do is first you wanted to split that. Okay, that splitting job is going to be done by your helicase. So it is going to break your DNA strand. It's going to break your DNA strand. That is going to be number 1, helicase. And number 2, you have something called as single stranded binding protein. Okay, so what it does is this is going to separate your, okay, it's going to be separating like this. Okay, so first you are going to have your helicase. This helicase, what it does is it is going to split your DNA. It is going to split your both the fragments of your DNA. So both the DNA strand is splitted. After it is going to split, there is going to be something called as SSB binding protein. What it does is this is going to bind to your adjacent both the ends of your single stranded DNAs, so that they won't go back and they won't bind together. So that is going to be your single stranded binding protein. So third one is going to be your primase. What is this primase? Primase is the one that codes for your primer, that codes for your primer, Primer. So what is this primer? So primer is the one that is going to, what it does usually is, the primer is the one that is going to help in, that is going to help in formation, starting the session. So it is going to start the formation of your DNA. It is going to start the formation of your DNA synthesis. So that is going to be your primase. After your primase, fourth one is going to be your topoisomerase. What is this topoisomerase? Topoisomerase is the one that is going to remove the positive and negative supercoils. It is going to remove the positive and negative supercoil. And fifth one is going to be DNA polymerase. So once it is going to separate, what it does is polymerase. What it does is, it is now you have it is separated. When a primer is started, this primer helps you for creating the area for action of your DNA polymerase. You have already removed the positive and negative supercoil by topoisomerase. Now there comes the action of your DNA polymerase. What it is going to do is, it is slowly going to move from this direction to this direction, and 3' to 5'. It is going to move from 3' to 5', and it is going to form the new DNA along with it. So you're going to form a new DNA along with it. Very good. This is easy. Form ஆயிடுது. But unfortunately, there is another strand which goes from 5' to 3'. அது என்னவாகும்? Let's see it next step. So first DNA polymerase, this is going to add nucleotide in the 5' to 3' direction. So 3' to 5' இது இருக்கு. அப்போ உங்களுக்கு இந்த இது எப்படி இருக்கும்? So new strand is going to be 5' to 3'. I already told you, right? So new strand will be 5' to 3'. So what it does is, it is going to add the nucleotide from 5' to 3' direction. அடுத்தது what happens is, இப்போ உங்களுக்கு டி இந்த மாதிரி வந்திருக்குல்ல, இப்போ இதை நம்ம என்ன பண்ணணும், ஸ்பீட் இன்க்ரீஸ் பண்ணணும். இந்த ப்ரோலிஃபரேஷன் ரெப்ளிகேஷன் ஸ்பீட் இன்க்ரீஸ் பண்ணணும். அது யார் பண்றாங்கன்னா, ஸ்லைடிங் டிஎன்ஏ clamps. They are going to increase the speed. And then you have your seventh one, that is going to be a DNA ligase, who is going to anneal your DNA. And finally you have your eighth one, that is going to be DNA polymerase. So what is this DNA polymerase? DNA polymerase 1 என்ன பண்ணுனா, they are going to seal the gap. So what is the seal the gap? So இப்போ cut பண்ணிட்டோம். So you're going to anneal, you're going to cut both the ends. So இப்போ both the end cut பண்ணா மட்டும் போதா, திருப்பி அதை join பண்ணணும்ல, அதுதான் DNA polymerase 1 பண்ண போகுது. And finally you're going to have your ninth point, that is going to be RNA. It is going to be synthetase. What it is going to do, it is going to remove those RNA polymerase. So you have some polymerase which is going to be there, right? So on the primers, RNA primers இருக்குல்ல, இந்த coding பண்ண primer, அதை ஃபைனலா remove பண்ணி எடுக்கிறோம். So again I repeat one by one. First you're going to have helicase, which is going to break your DNA strand. Then you have your single stranded binding protein. Then you have primase, RNA primers, which is going to code. Then you have topoisomerase, then your polymerase, then you have sliding RNA clamp, then the DNA clamp, then you're going to have DNA ligase, which is going to anneal your DNA. Then you have your DNA polymerase 1, which is going to seal the gap. And finally you have this, last but not least, the one that removes, the one that is going to remove your RNA primers, So for this, what you need to understand is, you have two strands, So this is your normal DNA. So normal DNA, it's going to have 3' to 5' and 5' to 3'. This 3' to 5' is called as leading strand, and this 5' to 3' you call it as lagging strand. One important problem which we have is, when the DNA polymerase is going to act here, it is going to move from 3' to 5', so no problem. There is going to be a continuous formation of your new DNA fragment. Okay, no continuous formation of your new DNA fragment, there won't be any problem. But unfortunately, what happens is here this is going to move in the 5' to 3' direction. If you want your DNA polymerase to move in the 5' to 3', it won't move. So what happens is your primers will be setting. If your primer, for example, if your primase is going to act at this particular point, your DNA polymerase will be moving in this direction. So what happens? First, it will start here. It will slowly move here. Then it is going to cut at this point. Then again, your primer is going to go and act here. Then you have your DNA polymerase. Then it is going to act till this point. So enna aguna break panni it is going to start. So in the way break panni start agumbodhu you have like this. So what you have is at last you have like this 1 2 3 in the my or broken fragments you will get. This broken fragment is called as your Okazaki fragments. Okay, Okazaki fragments. Okay, so this Okazaki fragments are discontinuous synthesis. So this is your lagging strand where you will have your discontinuous synthesis. So the ability of enzyme to catalyze many reactions before releasing your substrate is called possessivity. So in a leading strand, DNA polymerase 3 is going to act. So no problem, it is going to continuously form. இங்கேயும் DNA polymerase 3 இருக்கு. But unfortunately, what happens is, even though DNA polymerase 3 is there, you get some Okazaki fragments. So in the Okazaki fragments, enna pannanumna, again it has to. So what I wanted to do is, again you wanted to stick them together. So in the sticking them together irukku pathingala, இந்த வேலையை யார் பண்றாங்கன்னா polymerase 1 பண்றாங்க. Polymerase 1. So polymerase 3 is what you are going to do. Polymerase 3, actually they are going to do this leading fragment and lagging fragment. They are going to form the fragments. But leading fragments ஒரு single stranded வருது. அதே laggingல என்ன ஆகுது? Okazaki fragments. Okazaki fragments எல்லாரையும் ஒன்னு சேர்க்கணும். அதுதான் polymerase 1 ஓட வேலை. ஓகே, நான் இப்ப polymerase 1 வந்துருச்சு, seal the gap எல்லாமே பண்ணிட்டோம். இப்ப next நம்ம என்ன பண்ணணும்? What is the next ultimate thing? What we have to do is we have to know the fragments. We have to know the fragments and then we have to proofread it. So replication activity is going to be from 5' to 3'. Proofreading activity எப்படி இருக்கும்னா, proofreading activity is going to from 5' to 3'. So what happens is, just understand this, all the process happens in the 3' to 5' direction. So 3' to 5' direction, except your proofreading, which acts in the 5' to 3' direction. That is what I want you all to understand. Once you understand that, once you understand this Okazaki fragments and everything, next what you have to understand is, so in the Okazaki fragments எல்லாரையும் join பண்றது யாரோட வேலை? இந்த Okazaki fragments எல்லாரையும் join பண்றது தான் உங்களோட polymerase 1 ஓட வேலை. So now you understand all these steps, right? First, helicase. Helicase is going to break your DNA strand. Then you have your SSB protein. Then you have primase. Primase is going to primer it. Primer na paint adikimo primer adipom. Why? That is going to initiate the reaction. Then you have topoisomerase. Once the supercoil has been removed, then you have this DNA polymerase 3 producing fragments. Anga single fragment varum, inge multiple fragment varum. Then you have the sliding DNA clamps to initiate, to regulate the speed of your DNA polymerase. Then you have your DNA ligase, which is going to cut or which is going to annihilate. Then what happens? You're going to have your DNA polymerase 1, which is going to seal the gap. Then finally, the one to remove the RNA primers, you have this RNA. So, okay, RNA SE. Okay, so finally, this is over. So, and you understood what are the strands in your DNA, what are the strands in the replication. So, alpha part தான் the primase. Okay, alpha part is going to be your primase, beta part is going to make a DNA repair, and you have a gamma. What is this gamma? Stands for mitochondrial DNA production. So what is the difference between the our normal DNA and a mitochondrial DNA? Mitochondrial DNA edikoga heading mitochondrial DNA. What is this mitochondrial DNA actually? So nuclear DNA is a right-sided double helix. But mitochondrial DNA is closed and circular DNA. It is a closed and circular DNA. Okay, it has its own protein. It is going to have its own protein. It is going to have its own protein, tRNA, and genetic coding. Okay, so 100% it is inherited from mother. 100% it is inherited from mother. Okay, father வந்து contribute பண்ண மாட்டாங்க. Mother is the person who is going to contribute. Okay, yeah, why? Because so what happens is when the father's sperm is going to enter, so அந்த capacitization அப்படிங்கிற processல என்ன ஆகும்னா father-ஓட mitochondrial DNA is removed. Okay, before fertilization itself, father-ஓட mitochondrial DNA is removed. That is the reason why 100% mitochondrial DNA are inherited from your mother. இந்த mitochondrial இது இருக்கு பார்த்தீங்களா, all these are going to be helpful in development of brain, eyes and skeletal system. Brain, eyes, skeletal system இது மூணுமே inherited from the mother. Okay, so it is inherited from the mother, and because of that all the mitochondrial diseases, what are the mitochondrial diseases? So you have your brain, okay, in your eyes, in your skeletal system. Okay, this is called as Oxford syndrome. Okay, Oxford disease. Okay, you call it as Oxford disease. So it's a mitochondrial DNA. Also, it is going to be a reason for genetic disease of this. Okay, so you have this heteroplasmy. What is heteroplasmy? Do different types of mitochondria. So DNA rarely fathers can okay, what penetrate, and mother gives mitochondrial DNA. It is going to give mutational DNA. Everything is given by mother. Okay, and இந்த ஸ்டெப் எல்லாத்தையும் நம்ம ஸ்டாப் பண்ணணும். ஓகேவா, இந்த ஸ்டெப் எல்லாத்தையும் நம்ம ஸ்டாப் பண்ணணும். அதுக்கு நம்ம சில ஸ்டாப் கோடான்ஸ் இருக்கு. வாட் ஆர் த ஸ்டாப் கோடான்ஸ்? யூ நோ, உகா, உவா. What are the other things? I know you all know that. Just try to recall and tell me. So what are the different stop genes you know? What are the different stop codons you know? So uva, uga, uag, UAG. These are the stop codons. Okay, so இவங்க என்ன பண்றாங்கன்னா இந்த replication process stop பண்றாங்க. But unfortunately what happens, normal nuclear DNA la இந்த stop codons la work ஆகும். Okay, but what happens is in mitochondrial DNA there is some exceptions. There are some exceptions in the genetic code. Exceptions in genetic code. Okay, genetic code. So what are the exceptions you can find in the genetic code? In a normal, UGA is a stop codon in a normal in a nuclear DNA. But what happens in a mitochondrial DNA? It is going to give you tryptophan. It is going to give you tryptophan. Similarly, you have this AUA. AUA is going to produce isoleucine in a nuclear DNA, whereas inge it produces methionine. Inge it is going to produce methionine. Similarly, you're going to have your AGA and AGG. So this usually produces leucine, whereas inge vandu pathina, okay, inge vandu it is going to produce arginine. Okay, it's going to produce arginine, whereas in a mitochondrial DNA it is going to be a stop codon. So that means that all the mitochondrial as well as nuclear codons are going to be same except these three important things. Okay, so இப்போ DNA replication நம்ம பார்த்தோம்ல different steps. இப்போ இந்த different steps நம்ம inhibit பண்ணணும். எதுக்காக நம்ம inhibit பண்றோம்? ஒரு anti cancer agent. So இப்போ வந்து யூசுவலா பார்த்தீங்கன்னா anti cancer agents எல்லாம், so cancer எதனால ஏற்படுது? ஒரு unregulated DNA replicationனால தான் cancer ஏற்படுது, if I am right. Okay, so you all know that because of some unregulated DNA, because of the unregulated DNA only, so you are going to replication only, you are going to have a cancer. So இங்க நம்ம என்ன ஆகுது அப்படின்னு பாத்தீங்கன்னா, ஒரு anti cancer drug, it is going to target these specific sites. For example, human topoisomerase. So what I can do is I can target this human topoisomerase enzyme. So human topoisomerase na target panni na block pantena, that will be leading to a that will be preventing your DNA replication. For example, in the phase lan pathina, you will be blocking in the G2 phase pakana. G2 phase will block பண்ணுவீங்க. So what are the examples you can quote for this? Etoposide, adriamycin, doxorubicin. Just write along with me. Etoposide, adriamycin, and you have your doxorubicin. rubicin. So these are the three drugs which you are going to use: etoposide, adriamycin, doxorubicin. இவங்க மூணு பேர் என்ன பண்றாங்க, G2 phaseல topoisomerase block பண்றாங்க. Similarly, DNA polymerase. So you have your DNA polymerase, right? So you can block this DNA polymerase. How do you block this DNA polymerase? Again, similarly, you have your one particular drug called as 6-mercaptopurine. 6-mercaptopurine. 6-mercaptopurine என்ன பண்ணும்? It is going to inhibit your DNA synthesis. Okay, அதே மாதிரியே இன்னொரு ஸ்டெப் இருக்கு. இந்த DNA polymerase, இந்த topoisomerase-க்குலாம் function ஆகணும்னா what is required? You need nucleotide base, isn't it? So nucleotides இருந்தாதான் DNA replication நடக்கும். அப்ப இந்த nucleotide base வந்து யார் கொண்டு வராங்க? thymidine, guanosine, cytosine, okay, adenosine, guanosine, cytosine, thiamine or thymidine, இவங்கெல்லாம் required. அப்ப நம்ம என்ன பண்றோம்னா இந்த thymidylate synthase inhibit பண்றோம். That is what we spoke yesterday. What is the drug that is going to be acting on thymidylate synthetase? 5-fluorouracil. So there is other drug called as 5-fluorouracil which is used to block the DNA replication. That is going to be your human DNA replication inhibitors. You have certain drugs which is going to be targeted to the antibacterial. So நீங்க கேட்கலாம், என்ன எதுக்காக நான் ப்ரோகாரியோட்ஸ் பத்தியும் படிக்கணும், எதுக்காக நான் யூகாரியோட்ஸ் பத்தியும் படிக்கணும்? ப்ரோகாரியோட்ஸ்ல DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. ஸோ இந்த DNA polymerase 1, 2, 3 3 இருக்கு. ஸோ ஹியூமன்ஸ்ல இதுதான் வந்து பார்த்தீங்கன்னா it is going to function for the lagging phase. ஸோ லேகிங் ஃபேஸ்க்கு இவங்க தான் ஃபங்க்ஷன் ஆக போறாங்க. லீடிங் ஃபேஸ்க்கு யார் ஃபங்க்ஷன் ஆக போறாங்க? இந்த மாதிரி நம்ம வந்து செப்பரேட்டா we are reading for eukaryote and your prokaryote. Okay, so eukaryote, prokaryote, your polymerase 1 and polymerase 3. Polymerase 1 is going to go for the Okazaki fragments. Polymerase 3 is going to go for the normal DNA replication. But humans, delta is the one that is going to help both in leading strand as well as lagging strand as well as in Okazaki fragments. So, and the difference தெரிஞ்சுக்கிறது எதுக்காக அப்படின்னு பார்த்தீங்கன்னா, for a simple reason, you wanted to inhibit the bacterial replication whenever you are going to get a bacterial disease. Whenever you are going to have a bacterial disease, what you are going to have? You are going to inhibit. So how do you inhibit it? So you in the prokaryotes, the topoisomerase is called as gyrase. So what happens in prokaryotes? Okay, in prokaryotes, this DNA polymerase, this DNA topoisomerase, this topoisomerase is called as, what is that? Anyone? DNA gyrase. Okay, so you are going to block this DNA gyrase with the help of certain drugs, which all you know commonly. One is going to be your ciprofloxacin. Okay, other one is going to be your nalidixic acid, and other one is going to be your novobiocin. So all these drugs you will be using. Okay, the inhibitors of your DNA replication. Now, another application of this DNA replication which you have to know is, I talked about the mitochondrial DNA replication, isn't it? So this mitochondrial DNA is very important, that the problem in the mitochondrial DNA replication, it can actually lead to certain syndromes that is called as your Oxford's disease. So what are the three different Oxford's disease? You know, number one, Leber's hereditary neuropathy. Okay, Leber's hereditary neuropathy. So that is the one which you have to remember. This movie which came in Tamil, that is your Kanne Kalaimane movie. How many of you remember this Kanne Kalaimane movie acted by our Minister of Youth and Sports, Mr. Udayanidhi Stalin? How many of you saw it? Yes. So I'll show you that movie. See here. So you can actually understand it. So this is a movie came in Tamil, Kanne Kalaimane. So it was acted by our Honorable Minister Udayanidhi Stalin. So in that, he would have acted as a hero, and he would have been loving a girl, that is Tamanna. And after his marriage, what happens is slowly Tamanna will be starting to losing her eyesight. So when they take her to doctor, they will be telling that it is a mitochondrial disease. It is a rarest mitochondrial disease. And this rarest mitochondrial disease, how it come, if they once they started to analyze, that was when she found that her mother is also having a similar problem and mother is also having a blindness. So amma blind ah irpanga, adunala dhaan tamannavum blind ஆகுவாங்க. So acquired disease, கொஞ்ச நாள் கழிச்சு the mother, the patient or the person, the daughter will be like starting to lose her sight. So இது இந்த disease என்ன அப்படின்னு அந்த படத்துல என்ன சொல்லுவாங்கன்னா Leber's hereditary neuropathy அப்படின்னு நம்ம சொல்லுவாங்க. Okay, I hope so you will never forget this. So what is Leber's hereditary neuropathy? What happens basically in Leber's hereditary neuropathy is, this is actually a complex 1 defect. This is actually a complex 1 defect. Okay, it's a complex 1 defect, and the blindness will be cardiac blindness will be there, and cardiac conduction defect will be there. So blindness, along with that you will have cardiac conduction defect. Second thing is going to be myoclonic epilepsy, ragged red fiber. Okay, so disease, ragged red fiber disease. What is this myoclonic ragged red fiber disease? This is going to lead to myoclonic epilepsy. This is going to lead to myoclonic epilepsy. It is going to lead to myopathy. It is going to lead to seizures. This is going to lead to seizures, and this is going to lead to dementia. This is again one of the syndrome, one of this Oxford syndrome. And you have next one, that is going to be MELAS. What is MELAS? Mitochondrial encephalopathy. Okay, LA stands for lactic acidosis. You have lactic acidosis. Stroke, S stands for stroke-like episode. So this is your MELAS, and this also a complex 1 defect with lactic acidosis, strokes, myopathy, seizure, and dementia. Then you have the last but not least, you are going to have this Leigh syndrome. What is Leigh syndrome? Leigh syndrome is nothing but NDUFS gene defect. There will be movement disorder. So lactic acidosis occurs in all the syndromes associated with skeletal muscle. Remember, lactic acidosis occurs in all the syndromes that are associated with skeletal muscles. It occurs in your Leigh's, it occurs in your MELAS. It is going to be occurring even in the myoclonic epilepsy. So these 3 syndromes are going to be associated with the skeletal muscle. Okay, this is what I want you all to remember, and I hope so you will never forget. The last topic of discussion is going to be your DNA repair mechanism. So what happens is, so this is the most commonly asked question in your any set. So what happens is, there is a DNA repair mechanism that are present inherently in the prokaryotes and your eukaryotes. They are going to act like a checkpoint. So what happens is, there is going to be a mismatch repair. There is going to be a base excision can be occurring, or either it can go for a nucleotide excision or direct repair. So what happens? You're going to have a double strand, single stranded DNA. So single stranded DNA la edavadhu defect irundaduna, what it is going to do is, it is going to repair it either by mismatch repair mechanism, base excision repair mechanism, nucleotide excision repair mechanism, or direct repair mechanism. Either double standard repair mechanism, like you're going to have homologous end recombinant repair or non-homologous recombinant end repair. Okay, so that is done by your ERT, or you're going to have your NHER and NHEJ. So I'll tell you accordingly to this. Clear paranga. So you're going to have this repair enzyme can distinguish the parent and daughter DNA strand. Okay, so no new strand irukku, but if paranga, the new strand is not having methylation. So parent strand la methylation irukku, but new strand la methylation illa. Okay, C the parent strand, you're going to have T. Okay, mutation, methylation illa. Ada yaar kandupidikiranga mute S, MUTS. So mute S, what it does is, it is going to recognize the mismatch and they are going to bind at that particular area. So if a bind mute L, mute S, they are going to bind together and form a complex. Then this mute L activates the endonuclease in the mute H. இந்த endonuclease என்ன பண்ணும்னா, okay, this endonuclease is going to bind it. Then followed by the endonuclease, what you have is your H. You are going to have your exonuclease. This exonuclease and helicase, what it does is, they are going to cut the portion. They are going to cut the portion. So what they are going to do, in the portion in the portion cut பண்ணிடுறாங்க. In the portion cut பண்ணிட்டு next என்ன பண்றோம், once after this these two portions, what we are going to do is, we are going to have this DNA polymerase 1 or 3, which is going to come again, and they are going to synthesize a new strand. Once they synthesize new strand again, what they will do, there is DNA ligase comes, they are going to unyield the ends. Then you have your again a repair mechanism to check whether the problem is damaged, set is going to be made clear. Okay, so base excision repair mechanism is done to remove and replace the modified bases. DNA glycosyl bases binds DNA and removes the damaged gene. So AP endonuclease, what is that? Upnuric, then you have a pyrimidinic nuclease. So then this comes your DNA polymerase, which will be filling the gap. Okay, this is how you have your nucleotide excision repair. So you have your base excision repair. So base excision na ஒரே ஒரு base problem இருந்தா, then you have this nucleotide excision repair. ஒரு full nucleotide அந்த 10 இதையுமே கட் பண்ணிட்டு again இந்த 10 இதையுமே கட் பண்ணிட்டு new undamaged DNA is replicated. And this is what your xerodermopigmentosum. So what is going to be this xerodermopigmentosum? Why you need to understand this? Okay, why you need to understand this? You need to understand this because one of the most frequent defects in the xeroderma pigmentosum is autosomal recessive genetic defect in which nucleotide excision repair, that is going to be NER. I told you, right? This NER is mutated, leading to reduction or elimination of your NER. Because of elimination of NER, there is going to be no mismatch protein, repair. So if there is no mismatch repair, what happens? You will be having a mutated gene. Mutated gene causes xerodermopigmentosa. This comes to the end of a very important topic, that is going to be your DNA replication. Once again, I will tell you the DNA replication in a short and sweet manner. So first you are going to have two things. Either it's going to be prokaryote or eukaryote. Prokaryote, origin of replication is going to be at one particular site. In the eukaryote, it is going to be at the multiple site. Okay, so replication fork start ஆகுது 3-5. So first what happens, there is going to be a fork வந்து நம்ம பிரிக்கணும். So பிரிக்கிறது யார் பண்றாங்க, பிரிக்கிறத பண்றதுக்கு பேர் வந்து DNA helicase. So DNA helicase பிரிக்க ஸ்டார்ட் பண்றாங்க. Then அதுக்கப்புறம் you have this SSB binding proteins which is going to separate them. Then you are going to have your primase. Primase is the one that is going to help in initiating. Then you have topoisomerase. This topoisomerase is the one that is going to remove the supercoils. Then you are going to have your DNA polymerase that is going to come. The DNA polymerase on the one, you have your clamps, what that is going to fasten your DNA polymerase activity. That is going to produce from 5' to 3'. It is going to produce a new strand from 5' to 3' in the direction of 3' to 5'. Then what happens? You are going to have your DNA helicase. So DNA helicase எல்லாமே முடிஞ்சதுக்கு அப்புறம் you have your DNA ligase. This DNA ligase, what it does, it is going to பாருங்க இதான் உங்க helicase. So helicase ஓட வேலை என்ன, strands பிரிச்சு விடுறது. அதுக்கப்புறம் you have your primase, it is going to help in starting. Then you have RNA primer. First இது வந்து RNA primer தான் கொடுக்கும். Okay, RNA primer ஓட வேலை என்ன, first nucleotide base வந்து base pair வந்து RNA primer கொடுக்கும். அந்த RNA primer கொடுக்குற அந்த base pairல இருந்து தான் உங்களோட RNA polymerase ஓட work ஸ்டார்ட் ஆகும். RNA polymerase continuously போயிட்டே இருக்கும். What happens is, you will be having a new strand formation, if I am right. So you have a new strand formation, if I am right. Then what happens is, you are going to slowly it's going to form a new one. So on the new one form ஆயிட்டே வரும்போது, you have your DNA polymerase, and at the end of it, what happens, there is going to be action of DNA ligase that is going to help in the formation of your DNA ligase, which is going to cause annihilation of your DNA. And then you have your DNA polymerase 1 again, which is going to fill the gap. Okay, wow. So இதான் உங்க topoisomerase. Can you see? This is your topoisomerase. So that topoisomerase is going to remove the supercoiling. Supercoil remove பண்ணி விட்டாதான் உங்க helicase வந்து DNA strand remove பண்ண முடியும். So அதுதான் அதோட வேலை. அண்ட் இன்னும் ஒரு சிம்பிளான இது என்னன்னா லீடிங் ஸ்ட்ராண்ட் ஸ்ட்ரெயிட்டா இருக்கும். அதே வந்து ஆர்என்ஏ பிரைமர் ஒவ்வொரு தடவையும் வந்து உட்காந்து ஒக்கசாக்கி ஃப்ராக்மெண்ட்ஸ் கிரியேட் பண்ணணும். அதுதான் வந்து இதுல ப்ராப்ளம். அண்ட் டெர்மினேஷன் யார் பண்றாங்க? டெர்ப் பைண்டிங் புரோட்டீன் பைண்ட்ஸ் டு த டெர்மினேஷன் சீக்வன்ஸ். ஸோ டெர்ப் பைண்டிங் புரோட்டீன் தான் வந்து டெர்மினேஷன் சீக்வன்ஸ்ல உட்காந்து பிரிக்கிறாங்க. அதுக்கப்புறம் யூ ஹேவ் யோர் டிஎன்ஏ ப்ரூஃப் ரீடிங். ஸோ திஸ் இஸ் த என்டையர் சம்மரி. இந்த இதை வந்து ஸ்கிரீன்ஷாட் எடுத்துக்கோங்க. This is the entire summary of your DNA replication. I hope so you're clear with the DNA replication. If you have any doubt in the DNA replication, you can contact me. I'll be happy to solve you. In the next session, what we will be going is, we will start with 7 MCQs. After completing those 7 MCQ, we will go for the DNA repair mechanism. இன்னும் கொஞ்சம் டீடைல்டா நம்ம DNA repair mechanism பார்ப்போம். அதை பார்த்துட்டு நம்ம அதோட disease associatedையும் பார்த்துட்டு நம்ம transcription, gene expression and translation we will see in the next session. Okay, thank you.